Okay, so Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay, and a very good afternoon to all my beloved S1CP1 students. Alright, so today we are going to discuss our tutorial today. Okay, topic. Okay, topic today is about 8.3. Okay, and as well as we have planned, okay, group 3 will continue presenting about the cDNA, right? So before we can proceed, teacher would like you to sign up your attendance dulu, eh? Alright, so allow me to share my slide. Okay, can you see my slide, class? Okay, so teacher would like you to sign up attendance, our attendance first for today's class. Okay, so today we are going to learn about chapter 8.3 and we are going to continue, okay, one topic that known as application of recombinant DNA technology. Okay, recombinant DNA technology in gene cloning. Right, so 8.3 is the topic of application of DNA, recombinant DNA technology, and especially our focus is on synthesis of cDNA. Okay, so kita ini akan fokus on steps in production of cDNA, and after you have understand the process, then you are going to apply your understanding, okay, to go through how this process of production of cDNA able for scientists, okay, to produce artificial insulin in order to help those which has diabetes. Okay, so have you done scan? Okay. Just now Aimi kata tak boleh scan. Okay, so maybe blocked by the comment just now, right? So teacher show again the QR code. Okay, so that you can scan the QR code. Okay, class, Okay, so fill in your names, your metric number, right? Your session today, and of course your emotion, Okay, hoping everybody in good condition. Okay, always stay at home, stay safe. Okay, and teacher also happy for all our students, like Suhui already safely arrived. Okay, Suhui, lega dah sampai. Okay, alright. So have you done with that, class? Okay, so group 3, who are going to present your slide? Ratika, Togai, Suhui, Umi, Umu, Zinglan, and we have apa ni? Special invitation, Okay, from Yogin. Okay, Yogin ready, nanti nak nyanyi lagu Empat Dara, Okay, so teacher stop sharing, right? Now back to okay, so siapa nak present group 3? Umu. Okay, Umu. So Umu you can share your screen. Those yang nak tanya, you can type on the chat kelas below.
Kan dia tak boleh tekan share. Tak boleh nak tekan share.
Tak boleh tekan share. Oh. Okay, orang lain boleh tak cuba? Ataupun Umu forward kat teacher? Sebab yesterday when teacher buat dengan Umi belakang stage Umi ada? Okay. Semalam teacher buat dengan Nani. Nani turn on your camera. Because yesterday teacher buat dengan S1CP2, dia boleh share. Dia punya feature tu Umu tak jumpa
Ada, tapi kalau nak tekan share tu tak boleh. Dia jadi boleh tekan cancel je.
Cancel. Okay, anyone from group members yang lain? Do you have any? So that means tak semestinya Umu je yang share. Orang lain pun boleh share. Okay, do you have the copy? If you have the copy, then you can send to teacher. Umu? Okay. Sambil tunggu Umu setting her slide. Yogi nyanyilah boleh.
Cikgu last, cikgu.
Last. Oh, last. Okay. Panjang sikit lah.
Tak. First time dengar, cikgu.
Sebab Umu tak boleh share ni, macam mana?
First time saya
Okay,
dengar.
kalau tak boleh share you punya slide, then teacher open my one, then you present lah Boleh share tak?
Tak boleh.
Tak boleh juga.
Sekejap, Rati try.
Okay. Okay, Ratika, boleh? Ratika, okay tak, Ratika? Okay, kalau tak boleh, then teacher share my slide and then you present lah Okay?
Dia boleh briefing cikgu tapi tak nampak.
Tak nampak? Apa maksud apa tak nampak?
Sudah
Ratika kena tekan share screen. Okay, entire screen. You choose the entire screen. The entire screen. Boleh?
Nampak tak cikgu?
Tak ada lagi.
Ada tak?
Tak ada lagi. Tak letak. Nampak?
Dia tak tunjuk cikgu. Saya dah share screen dah.
Okay. Tak apa. Kalau tentu you all guna slide cikgu je lah to explain your understanding, okay?
Ada dekat
Okay.
tele cikgu.
So, let's start. Tak nak. Oh, dekat tele? Oh, kamu mau hantar ke? Kat tele siapa? Saya tak jumpa. Tele Atika ke?
Sekejap kita rasa. Tak, dekat tele group Bayu.
Tele okay, Suhui dah hantar. Alright, okay. So, teacher buka lah yang Suhui punya. Suhui. Okay, so just get ready and teacher share your PowerPoint ni, then you present Okay, so class, we are going to hear from group Suhui and the gang. Okay, to present about the cDNA. Okay, okay, so right, so teacher invite group 3 to present your understanding about what is cDNA. Okay, so who are going to be start? Who want to start the presentation?
Umur.
Umur. Okay, go ahead, Umur.
Today we're going to talk about cDNA. So cDNA is actually C is stand for complementary DNA. Cikgu next.
Wait. Your friend boleh nampak tak? Wait. Oh, dalam YouTube dia macam lambat sikit lah kan? Okay, YouTube, right. Okay, next. Right, so the cDNA, yes.
cDNA is complementary DNA. DNA molecule is synthesis in vitro using mRNA template catalyzed by enzyme reverse
Okay, Umur, stop.
transcriptase.
Okay?
So, DNA molecule synthesis in vitro using mRNA template catalyzed by enzyme reverse transcriptase.
Okay, so in this process, kita nak belajar apa ni, Umur? The process of? The process of? Can't hear you, Umur?
Production of cDNA.
Wait, Okay, production ataupun synthesis of in vitro using
mRNA template.
Okay, mRNA template Wait teacher nak tukar layout sekejap. Let me presenter sebelah Okay, bagi nampak kamu sikit Okay tak? Okay, so you can continue. Okay, so DNA molecule synthesis in vitro using mRNA template. Okay, kelas pertama sekali in vitro penting. In vitro Number 2 is mRNA template. Okay, number 3 apa Umur?
Enzyme reverse transcriptase.
Okay, number 3 is enzyme reverse transcriptase. These are all the important when we talk about cDNA, about complementary DNA. Okay, continue.
cDNA molecule is identical to a native DNA but lack of non-coding region which is introns. means cDNA need lack of introns. Then the major source of DNA, which is insert into vector and be cloned. cDNA made in laboratory from isolated DNA. cDNA method, they produce limited kind of gene library, which is a cDNA library. It also represent cDNA library only represent part of cell genome because it only contain exon, the gene that we transcribe. Therefore, cDNA molecule correspond to a gene, but lack of intron present the DNA of the genome.
Okay. So your your slide is so informative. But teacher would like Umu to recap balik. Okay, in order for your friends to understand this complementary DNA, okay, apa beza DNA, the molecule of DNA in the nucleus. Okay, when we mention about DNA, okay, we always, okay, kalau eukaryote is in the nucleus lah, okay, kalau prokaryote is the nucleoid region. Okay, apa beza, okay, DNA versus cDNA. Okay, DNA versus cDNA. DNA ni dalam nucleus or nucleoid. Okay, cDNA is in vitro. Okay, apa maksud in vitro? Umu, is a test tube. Okay, macam apa ni, bayu tabung uji IVF Okay, in vitro apa tu, bayi tabung uji tu sebenarnya adalah konsep menghasilkan apa embryo or the zygote or the embryo in vitro, not inside the womb of the woman atau of the mom, right? So same goes here. So DNA versus cDNA. Jadi what is the main difference yang teacher nak Umu highlightkan pada kawan? Umu? What is the main difference, Umu?
cDNA single stranded.
Single stranded tu letter dia akan lagi double juga. Itu proses ni yang paling ni ada jawapan ni cikgu. Okay, teacher highlightkan. The main difference DNA versus cDNA is yes, intron. Okay, previously during chapter 6 you all dah belajar, right? Okay, before after complete the transcription, okay, kelas the central dogma. Okay, cuba sebut balik Ratika konsep of central dogma. Konsep of central dogma, Ratika, from chapter 6 DNA. Hello Ratika. Ratika, are you there?
Central dogma is a framework for understanding the flow of genetic information between DNA, RNA and protein in living organism.
Okay, itu dia punya definition lah. But teacher nak you summarizekan the concept so that can apply here. Okay, DNA. Okay, what is the process, Ratika?
Perfect DNA replication.
Hai, replication tu apa tu? Kita nak DNA to mRNA, we call it
Transcription.
Yes, transcription. Okay, transcription DNA to mRNA is a transcription, right? So we are looking at this stage. Class That's why when you search a diagram to support your concept under central dogma, kan cikgu tegur you all perasan tak nanti ada satu arrow yang dia boleh buat patah balik ke DNA. Those yang dapat gambar macam ni betul tak Nabila? Okay, dia ada satu there is a certain diagram yang akan tunjuk mRNA boleh patah balik jadi DNA. So this is where you apply the concept. Okay, so what is the name of method to produce cDNA? Apa nama prosesnya? Okay, cuba tanya kawan kat luar. Atika tanya. Name the process.
Name the process of cDNA.
To form cDNA. Apa nama prosesnya? Okey, panggil cikgu dah bagi jawapan tu. Okey, tengok. Okey, cikgu tutup sekejap. Kita balik pada konti kita. Okey, mula Wan jawab Wan Okey, Wan jawab Wan. What is cDNA Wan? Type kat situ Wan. Wan type kat ruangan chat What is cDNA? Wan jawab Okay, what is cDNA? Teacher nak Wan jawab. Wan. The concept of cDNA Okay, show. Nampak tak show? Okay, what is it? Wan, what is cDNA? Wan jawab kat chat ruangan chat Wan. Okay, Wan. Okay. And then teacher nak Udin Okay, Udin name name the process to produce cDNA. Okay, name the process that produce cDNA. So teacher nak tu Wan jawab tu Wan jawab and Udin jawab yang dua. Okay, what name the process that produce cDNA. Okay, so ruang chat tu tolong jawabkan. Okay, now kita continue balik kepada umur. Right, so like teacher mentioned just now, okay, in vitro means apa kelas? Made in the laboratory. Okay, how we isolated the DNA. Okay, so teacher would like you to bear in mind the concept of cDNA library. Okay, cDNA library ni means apa umur?
Yeah.
What is the meaning of cDNA library? Okay, kalau teacher mention library, you all boleh bayangkan perpustakaan. Okay, and always when mention about library, related to books. Am I right class Related to books. Okay, but now tiba-tiba we use this term of library. Kita pinjam kita pinjam term library ini in DNA technology and we label is a cDNA library. So what you understand about cDNA library? Tak semestinya umur kawan-kawan lain, the group member boleh tolong juga. The group member, okay, when you prepare this concept, okay, what do you understand about the cDNA library? Wong? Any idea about the cDNA library? Hello? cDNA library means this, it caps apa? Tengok kat situ Okay, kalau kamu ayat kamu kat sini. Apa bunyi ayatnya kat situ? Methods that produce only kind of live gene Kind of gene. That means cDNA ni not represent the whole DNA gene. Boleh faham tak kelas? Not represented the whole DNA gene. You all dah belajar the concept of DNA. Am I right kelas Okay, DNA carry all the genetic information. Am I right? Okay, so when this genetic information, it will carry all sorts of gene But in cDNA, we only select certain part of cDNA. Okay, certain part of cDNA which we consider as the first tool that we have learned, iaitu what we call as a target DNA. That is what this cDNA apply in the concept. Okay, right, cDNA library. Okay, next. Right, we're going to continue the process. Umu, have you done your job?
Yeah, lagi.
Okay, so can you see my slide, class?
No.
Can you see my slide?
Tak.
Okay, so teacher dah share your slide, your second slide.
So now, next is the production of cDNA. mRNA extract from the cell. Enzyme reverse transcriptase is used in vitro to make single-stranded DNA. So means that the enzyme from double-stranded becomes single-stranded DNA. And then enzymatic degradation of mRNA, the second DNA strand, complementary to the first, is synthesis by DNA polymerase. So then single-stranded DNA polymerase akan jadikan benda tu double-stranded. Double-stranded DNA is modified by addition of restriction enzyme recognition sequence at each end. cDNA insert into vector DNA in manner similar to insertion of genomic DNA fragment.
Wait. Okay, do not read from your slide. Understand the process, then you will able to explain it better. Okay, who's explaining? Umi Is it Umi? Still Umu? Oh, still Umu. Okay, tengok jap kita tengok Wan jawab betul tak tu Atika? Kita komen jawapan Wan dulu. Wan jawab cDNA is a complementary DNA. Is it correct?
Correct.
Yes, tahniah Wan Okay, very good. Nanti kita belanja apa tu lagu Empat Dara from Yogin from our singer, top singer of S1CP1. Okay? Where's Yogin? I can't see him. Where's Yogin? Yogin, where are you? Oh, you backstage Yogin? Yogin's backstage already. Okay, so kita yes, good one. Right, so then teacher asked just now, Uden. Okay, Uden Okay, so very good. Now teacher hide and then we may continue. Okay, so back to our sharing session. Okay, teacher share and okay, proceed. Okay, we may proceed. Production of cDNA. Okay, first kena ada apa? How to produce cDNA? First, what you must have?
mRNA.
Yes, first we must have the mRNA as a template. Okay, so how to produce or to synthesis the DNA? Okay, what enzyme? Name the enzyme that use?
Enzyme reverse transcriptase.
Yes, enzyme reverse transcriptase. Alright. So when mRNA or okay, how to produce DNA class? What enzyme that we use to produce the mRNA, DNA? The second DNA?
DNA polymerase.
Yes, DNA polymerase. Okay, so what happened to the mRNA? What happened to the mRNA just now as a template? Ratika, what happened to the mRNA just now as a template? Just now mRNA as a template, right? So we have mRNA, right? Am I right, class? Okay, mRNA. And then with catalyzed by enzyme reverse transcriptase, we produce the single cDNA. Am I right? Okay, so this single cDNA with catalyzed by DNA polymerase, it will produce another strand of DNA, another strand of cDNA. Okay, so the yellow one is the first cDNA. The purple one is the new cDNA that catalyzed by DNA polymerase. Okay, DNA polymerase. So it become double-stranded DNA complementary. Okay, cDNA. So this cDNA Okay, all already have double stranded. One yellow, one purple. Okay, yellow is from the first single stranded. Okay, first single strand where transcript from mRNA, and then this single strand. What happened to the mRNA just now, class? It will undergo degradation. Right? Okay. So double stranded DNA nanti macam mana nak apa tu nak continue to produce insulin? How we going to produce the atau nak multiply the cDNA? So kita nak multiply the cDNA tu, guys?
By addition of restriction enzyme.
Itu kalau kita nak is modified enzyme. Okay, now we want to amplify. Nak make many copies of the cDNA. So again, we follow the step. We will insert this to a carrier of the cDNA, which is known as vector. Okay, similar like what we have discovered under gene cloning. Okay, so it's a manner similar to insertion of genomic DNA fragment like we have done under gene cloning. Okay, so next, siapa lagi nak sambung? Okay, next.
And then cDNA that are cloned make up a library containing a collection of genes.
Okay.
So,
siapa nak present ni? This one nak ke?
this one is,
Hello?
yeah.
This diagram is actually exactly from our books, Ratika? Exactly from our notebooks where showing the for cDNA formation. Do you want to go through? Or we can skip this? Skip, alright? Okay, next we go to the last slide. Am I right?
Yes.
Okay. So who's presenting?
Umum.
Can't hear you, my dear.
Umum.
Umum juga? Oh, hari ni umum buat semua. Pukul habis. Alright.
The production of insulin using cDNA and E. coli. First transcription process will produce mRNA code for insulin that extract from pancreas cell. Reverse transcriptase enzyme catalyze reverse synthesis of cDNA from mRNA template. Then MR template will discard by using mRNA degrading enzyme. And then DNA polymerase used to synthesis the second cDNA strand. And then it will produce required gene which is coding with insulin in cDNA double-stranded. Next, restriction enzyme, it used to cut the cDNA at specific site and create the sticky end. The plasmid vector from an E. coli is removed and open up a specific point using the same restriction enzyme. Next, cDNA are insert into the open plasmid and sealed by using the DNA ligase to form rDNA. And then rDNA will transform into host cell E. coli by process transformation and then screening to obtain clone E. coli. And then E. coli and recombinant plasmid allow to reproduce. Then a lot of bacteria clone carrying many copies of gene for insulin. Then bacteria can be used to produce much insulin for health industry.
Okay, done. Alright, so where is the different class? Okay, where is the between the gene cloning, the step in gene cloning, and the usage of cDNA in the gene cloning and E. coli. What is the role of E. coli kat sini, Wong? Wong, what is the what is the role of E. coli in this step? Hello, Wong? Wong, can you hear me? Hello, Wong? Hello, Wong? Can you hear me? Nabila, can you? Nabila, okay, Nabila is not group 3 Ratika lah, okay, Ratika. Okay, what is the role of E. coli in this situation?
Host cell.
Host cell, good. Host cell satu. Lagi satu? Selain pada host cell, Ratika? Okay, perasan tak? Where is, okay, first is a host cell, then satu lagi, E. coli ni memang kalau you, dia boleh jadi tokai bakteria. Okay, why? Teacher mentioned that because first, it will be as a host. Okay, see class here, where is the, okay, host cell. Host cell E. coli, host cell. One rules. Another rules, ni. Ratika baca ni. Another role, E. coli akan. Ratika nampak tak ni? Ratika yang cikgu highlightkan ni. Bulat E, sans, U. Yang kelima, Yang kelima, baca kan? Yes. Okay, E. coli. Okay, selain pada letter become a host, dia juga akan jadi sebagai vector. Okay, vector. Vector means apa kelas? That carry the cDNA. Okay, so E. coli will play two roles in production of insulin. Right, so first as a vector, where you get the plasmid, you extract the plasmid from the E. coli, right, and you will remove the remove at the palindromic sequence, okay, as the restriction site, and you open up, okay, this restriction site in order you to insert the cDNA inside the plasmid. Okay, so cDNA are inserted into the open plasmid, and as you all remember, okay, we have the modifying enzyme, okay, tools number 4, under gene cloning kita ada 5 tools. So tools number 4, DNA ligase will seal, okay, forming the phosphodiester bond, okay, in order to form the recombinant DNA. So meaning this recombinant DNA, right? So if teacher draw like this class, so the recombinant DNA will have, okay, what we call here class? So if teacher show this one, the red color here, when teacher sketch this one, class, so this will contain the cDNA, right? The cDNA that we have already inserted. Okay, so then we introduce this rDNA which carry the cDNA into the E. coli again as a host. Okay, that's why you all nampak kat sini kelas on the on the, okay, here. Okay, you can nampak kat sini. Okay, you juga must undergo the screening process. Okay, why we need to go the screening process? Because kita nak choose which E. coli bacteria that can or that up the recombinant plasmid in order kita nak amplify, amplification. Okay, amplify to make many copies of the cDNA which carry the gene of insulin, so we can use, or produce insulin as much as required, for healthy industry to help those yang diabetes. Okay, so that's all for from group 3. Okay, group 3, thank you for presenting about the cDNA. Okay, so now let's back to our, let's wait, teacher stop sharing. Okay. Okay, teacher would like you to teacher would like you to teacher would like to welcome your senior Akmal Nur Farahin, right? For joining us. Okay, joining us petang ni. Akmal Nur Farahin is actually your senior, your batch last session. Okay, so Wong is on mute. Wong is on mute, Wong Okay, alright, so where's Yogin? Yogin is here, is it? Okay, before we stop our today's class, okay, teacher would like to invite Yogin. Where's Yogin? Okay. So everybody, kita focus to Yogin, alright So Yogin So where's Yogin lah
Lalu,
teacher?
Boleh dengar cikgu?
Boleh. Very clear Yogin.
Boleh.
Lama sikit Yogin Okay. Alright lah, okay. Okay. Nobody at home, right?
Ada
orang
cikgu
ke?
bawah. Ada.
Your mom?
Semua ada.
Semua ada. Okay lah.
Hmm.
Okay, 1, 2, 3.
Lagu pada okay. Empat darah duduk di tepi sungai, duduk menjerat sekitar batu. 1, 2, 3, 4 anak darah. Semua orang tahu aku boleh mengira. Tapi kalau hilang satu anak darah, bikin risau satu negara. Semua anak darah, jangan mudah percaya janji sederhana. Jangan mudah terpedaya dengan dunia. Dan 4 darah
Duduk di tepi
sungai, duduk menjerat sekitar batu.
Masuk Ratika.
Masuk. Darah duduk di tepi sungai, duduk menjerat sekitar batu.
Masuk guys. Okay lah kelas thank you very thank you Yogin. Kelas bagilah kan, right? Thank you for
Thank you.
Okay, supportive petang ni. Entertain your friends. Wan kau jangan gelak kat pilah tu Okay, so I think for our session petang ni we end until here. Nabila, esok Nabila Newton Okay, so teacher kita jumpa lagi tomorrow morning. Okay, and we're going to continue discuss the question that teacher already shared with you. Okay, Yogin, nanti balik college Sekolah Pila, cikgu belanja Yogin. Okay, I think that's all for today class. Thank you. Okay, and hoping you always stay at home and make sure you complete the question so that tomorrow we can discuss, go through together. Okay, so any words from Yogin last before we end our live?
Last word apa cikgu? Okay, semua nyanyi cikgu. Semua nyanyi. Suruh semua buka mic.
Okay, Suhui, Umi, Ratika, Togesh, Nabila, Umu. Okay, Yogin ajak you all nyanyi, jom. Umur, Togesh, Nabila, Umur, Umi, Ratika, Suhui and cikgu sekali. Okay. Spotting lah petang-petang ni sebelum kita you stop. You have class after this, no right? Do you have any chemistry class? Today is Rabu kan? Ada chemistry class ke?
Ada practical test.
Oh, practical. Oi, ada practical. Okay lah. Okay, tak apalah again. Tomorrow lah kita continue. Okay, kamu simpan suara dulu simpan suara tomorrow test. Okay, alright. All the best class for your practical test. Okay, hoping you can answer it very well. Okay, so we meet again tomorrow. Okay, class, bye. Thank you. Nice to have you together with me live. So nanti next time kita invite your another friends pula. Okay, bye class.
Okay, cikgu.
All the best.
Bye.
Nabila, Togesh, Umu, and your Thank you. Bye.
Bye.
Okay.